We are going to talk about primary hemostasis in this video. We're going to discuss and describe the physical structure and functions of blood vessels within the hemostatic mechanism. We'll also discuss the role of platelets, including their structure and function. So overall, in this short video, we want to understand the mechanisms of the initial clotting system and how we maintain vascular integrity. Within our vasculature, blood flows through a closed system called the circulatory system. All of the vessels and constituents are critical in really controlling the integrity and functions of that system. When we have an injury that leads to a bleed, our body tries to minimize the blood loss by activating platelets and dissolved plasma proteins to form a clot, which occludes that injured vessel and prevents further blood loss. So really, we want to have the hemostatic mechanism activated where it is needed and only in that area of injury. We have a series of stages and surveillance systems that help keep things in control. So when we talk about hemostasis, we're really talking about the property of circulation that maintains blood in a fluid-like state within those blood vessels. And under normal circumstances, the ability to prevent excessive blood loss during injury. Blood coagulation is a mechanism that transforms that fluid plasma into a gel by converting the soluble protein fibrinogen into an insoluble form called fibrin. Hemostasis occurs in stages, first in primary, then secondary hemostasis, then finally fibrinolysis. During primary hemostasis, the platelets interact with the injured vessel and with one another. This interaction results in a mass of platelets known as the primary hemostatic plug, which temporarily stops bleeding, but it is fragile and it is easy to dislodge these platelets from the vessel wall. So next, insoluble strands of fibrin become deposited on and within this platelet plug to stabilize and reinforce that clot to allow wound healing and further blood loss. Fibrin is formed in a complex series of reactions involving coagulation factors, and this fibrin reinforced plug is called the secondary hemostatic plug or secondary hemostasis. Once that vessel is repaired, we also need to restore normal blood flow by lysing that clot. The process of removing fibrin is called fibrinolysis. In this system, we remove fibrin from the vascular system in a controlled manner once it has fulfilled that function, and it also helps protect excessive fibrin accumulation in that area. Hemostasis involves several systems working together to stop bleeding and repair blood vessels. First, we have the vascular system, in where blood vessels constrict or vasoconstrict to reduce blood flow and minimize bleeding. Next, we have the platelet system, and the platelets adhere to the injury site where they are activated and aggregate to form that temporary plug. Then we have a series of the coagulation system, which involves clotting factors and proteins and fibrin-forming proteins to convert fibrinogen into fibrin that stabilizes that plug. We also have a fibrinolytic system where once healing occurs, plasminogen is activated to break down that clot and restore normal blood flow. And then we have the immune system, which influences clotting through inflammation. Within hemostasis, the vascular role is really to vasoconstrict and initiate the hemostatic process. But let's talk about the vasculature system and what it entails. First, the key components of the vascular system includes arteries, where they carry oxygenated blood away from the heart to the tissues. They have thick muscular and elastic walls that allows them to withstand and regulate high pressure created by the heart's pumping action. The inner layer is lined with smooth endothelium, which helps reduce friction and maintain that blood flow. Veins carry deoxygenated blood from the tissues back to the heart, and they have thinner walls than arteries and larger lumens. They contain valves to prevent backflow of blood, ensuring it moves in one direction towards the heart. Capillaries are the smallest blood vessels where the exchange of gases, nutrients, and waste products occur between the blood and the tissues. They consist of a single layer of endothelial cells, and they allow easy diffusion of substances between blood and tissues. During an injury, blood vessels undergo a series of protective responses to protect excessive bleeding and promote healing. Immediately after injury, blood vessels constrict, or it's called vasoconstriction, to reduce blood flow and limit blood loss. The endothelial cells lining the vessels become activated, exposing underlying structures like collagen and von Willebrand factor, which helps attract platelets to the site. Then we know platelets adhere to those exposed areas, become activated, and aggregate to form that temporary platelet plug. Simultaneously, the coagulation cascade is triggered, leading to the formation of the fibrin clot that stabilizes the plug. And this sequence of vascular responses helps control bleeding and sets up the stage for tissue repair. During vascular injury, the endothelial cells have a specific function that triggers platelet activation, vasoconstriction, coagulation, but also inhibits fibrinolysis. So the exposure of those subendothelial collagen and tissue factor is what activates platelets, which adheres to von Willebrand factor and releases signals to recruit more platelets. The endothelial cells release endothelin, causes vasoconstriction to reduce blood flow, in addition to nitrous oxide. Tissue factor activates the coag cascade, converting fibrinogen to fibrin, stabilizing that clot, and then plasminogen activator inhibitor is released to inhibit fibrinolysis, ensuring that clot stability during tissue repair. Platelets are small anuclear cell fragments essential for hemostasis that are released into circulation from the bone marrow by megakaryocytes, and its production is regulated by a hormone called thrombopoietin. During vascular injury, they adhere to exposed collagen via von Willebrand factor that triggers a shape change from disc to spiky spheres that enhances their ability to bind. These now activated platelets release granules containing ADP, serotonin, and thromboxane A2, which promote further platelet recruitment and aggregation. This aggregation forms a stable platelet plug at the injury site. Platelets also provide a surface for coag factor activation, leading to fibrin formation, which stabilizes the plug and supports long-term clot integrity. Platelet ultrastructure is composed of distinct zones that support their role in hemostasis. First, let's include the peripheral zone. This contains plasma membrane with glycoprotein receptors like glycoprotein 1b and 2b/3a, which is crucial for adhesion and aggregation. The membrane is coated with a glycocalyx rich in glycoproteins that bind von Willebrand factor and fibrinogen. The structural zone contains a cytoskeleton network of actin filaments, microtubules, and contractile proteins like myosin. This structure maintains the discoid shape and facilitates the shape change during activation. The organelle zone houses granules essential for platelet function. So first, the alpha granules contain clotting factors like fibrinogen and von Willebrand factor, growth factor, and adhesion proteins. The dense granules store ADP, ATP, calcium, and serotonin, which are secreted to amplify platelet activation. Then the lysosomes contain enzymes that help in clot remodeling. The membranous zone includes the open conicular system, which increases the platelet surface area for secretion, and the dense tubular system, a calcium reservoir that activates platelet and granule release.